So good evening all and welcome back to Essential Academy. So till now we were discussing about in the previous lecture the active transport in which we have discussed about the P class type pumps in which we have discussed about the calcium 2 plus ATPases and Na plus K plus ATPases. So now next. So this is just the summary like how does your P class pump looks like in comparison to the F and V class pump, and then we have got the other one called the ABC superfamily where ABC stands for ATP binding cassette superfamily. So we'll all discuss these in detail slowly one at a time. So today we are going to talk about F-type ATPases. So remember once I have told you about the ATP synthase that it has got two domains F0 and F1. So that is the same today we are going to discuss in detail though, but so these F-type ATPases or F-type pumps, they possess transmembrane domain called F0, wherein I have mentioned that this 0 or O stands for oligomycin. Oligomycin, it's an inhibitor, and a peripheral domain called F1. Okay, so they are written like this: F0 and F1 particle. Now, they transport only protons down their electrochemical gradient for the synthesis of ATP. So till now, all the pumps what we have discussed, it actually causes ATP hydrolysis. Okay, this is the only pump in which we are going to talk about ATP synthesis, and that's why it is also called ATP synthesis. So here we are going to make ATP instead of use ATP. So the protons are used or transported down their electrochemical gradient and for the synthesis of ATP and from ADP and PI instead of hydrolysis, and hence they are called ATP synthesis. So in this we are going to talk about how ATPs are synthesized. Now where do you see ATP synthesis? Of course in the mitochondria, because that's what we say, right? Powerhouse of the cell is the mitochondria. So what is the power? ATP is the power. Who is creating this power? It is the ATP synthesis. Okay. Now, as already mentioned, F0 is an integral membrane protein complex. O indicated inhibition by the drug oligomycin. Please remember, many times I'm repeating because this is important. Now, it is a peripheral protein where one indicates that it was the first of the many factors which were isolated from the mitochondria. So, it uses the energy of ATP to transport the protons uphill. Okay. So, now we are going to study how these ATPs are synthesized. So, where all these ATP synthase or F pump is present in the bacterial plasma membrane, in the inner mitochondrial membrane and the thylakoid membrane of chloroplast. So once we start the biochemistry portion, there in which we are going to study that how mitochondria is actually, the complex. There is something called electron transport chain. Those who have started reading Leninger, please look to electron transport chain. Okay, very beautifully it is given in Leninger. So electron transport chain. So when the electrons are transferred, during that H plus are also H plus proton electrochemical gradient is also created, which results in ATP synthesis. Okay. Now, when do we study electron transport chain? After the glycolysis. So glycolysis we are going to study, then we are going to study TCA cycle, and after that comes your electron transport chain, in which we'll study how one glucose molecule is going to give you these many numbers of ATP molecules. Okay. So start reading now itself the Leninger which I have shared with you. Very beautiful it is given. And the enzymes also, it's very important. Then the thylakoid membrane of chloroplast. So we also know that the chloroplast is responsible for creating food, right? It is the food source, and the photosynthesis happens. To carry out the photosynthesis also, we need the proton electrochemical gradient. So these are the three where you can see the ATP synthase are present. Now look at the structure over here now. So you can see that, so among this, which one is going to be your F0 and which one is going to be your F1 particle? So this is, which is the transmembrane integral protein is your F0, and which is the peripheral is your F1. Okay. Now you can see that this F1 means it is present in the matrix. If we are talking about the mitochondria, then it is present on the inner membrane. So this is the inner membrane of your mitochondria. Okay, then the intermembrane space is there, and then here you can find the outer membrane. Okay, so this much is clear. So we have got the outer membrane, intermembrane space, inner membrane, matrix. On the inner membrane, you have got the F0 particle. In the matrix, you have got the peripheral protein F1 particle. Okay, now see the amount of the subunits present over here. Can you see over here? So we have got alpha, beta, trimer over here. Okay, so alpha, beta, alpha, beta, alpha, beta we have got. So it is like kept in alternate arrangement of this alpha, beta. Then you have got the delta and this B and alpha. So what you have to understand over here is that it is like a motor. Okay, so when H plus from intermembrane space to matrix it is going. So what is happening? H plus from intermembrane space to matrix, when it is passing through this channel, from here it is passing. So what is happening that time? It is generating a electrochemical gradient, right? Now this is sufficient to rotate your membrane. Okay, sorry, no, it is sufficient to rotate your ATP synthase. So what is happening? It is your rotating. Okay, now this rotation leads to ATP synthesis. This is little complicated, listen carefully. So I repeat once again. So what is happening? Here you have got H plus, a lot are there. Okay. What is happening? Electrochemical gradient is formed. Now this H plus needs to transfer from intermembrane space to the matrix. So while transferring here, the F0 particle is moving. It's like a motor, it is moving. Okay. Now this entire thing is rotated with the help of the gamma subunit. Okay, so this is the gamma subunit. It is rotating like this. Now this rotation is helping in the conversion of your ADP and PI to ATP. Is this much clear? Because I need to go in detail more. Can just someone tell me this much is clear to you?
Yes, ma'am.
Okay. Now what I want you to see is another figure, little bit more complicated. So slowly, I'm just building the concept. Okay. So you can see over here again, but it's a very beautiful concept. Okay. So you can see that you had over there the C subunits. How many are there? 10 are there. Okay. So that is your F0 particle I was talking about. Now, when this rotation is there, because of the movement of H plus, you can see how nicely this gamma subunit is rotating your alpha and beta. Okay. Now, what is the function of this A, B2 and delta? To hold the F0 and F1 particle together. Okay, clear? Now, how does the top view of this F1 particle looks like? So it looks like something like this. So you have got alpha, beta, ADP, alpha, beta, ADP, alpha, beta, ADP. Okay. Now, what you have to understand here is there are 3 sections, okay, of alpha and beta. Now, what you have to see is that first your alpha beta is empty. It means what? Nothing has been bound to that. Okay. Then what happens? Your ADP comes and binds to this alpha and beta. So it becomes your alpha beta ADP. Then when this rotates, okay, it gets the energy to become ATP. So this is called open, loose, and tight. Conformations of ATP synthase. Let me repeat once again. So first you have got these 6 subunits are there. 6 are divided into 3, alpha, beta, alpha, beta, alpha, beta, alternate arrangements we have. Now in the first case scenario, it is empty. then it gets bind to the ADP, and when the rotation happens because of the H plus proton gradient, this energy is utilized to convert ADP plus Pi to ATP. So what is happening now? So by the time this ATP is released, another ADP comes and binds. So what will happen? Then this will become open. In the next round, the empty becomes the loose becomes the tight. Are you getting the point over here? So let us say that one second. Let us say that this is first condition, this is second condition, and this is third condition. Okay. Now ADP comes and binds. ADP comes and binds. What will happen? This ADP is becoming your second condition, right? When it was empty, it was first condition. Now when ADP comes and binds, it becomes this one, right? So your first becomes second, correct? By that time, when the first becomes second, the second becomes third. So your ATP will be released. The moment ATP is released, it becomes your first because it becomes empty. So empty, loose, tight. Tight means ATP is there. Loose means ADP is there. Empty means nothing is there. So your open becomes loose, loose becomes tight. So all this rotates like this, like this, like this. Okay, is it clear, guys? Is it clear?
Yes, ma'am.
Okay. So there's one more figure, the same thing which just now I have explained. Okay, just so that you know your visualization becomes clear. So I got one more over here. So you can see that first we have, let us take from here. First we have the alpha beta ADP plus Pi. Okay, then what is happening? You get that ADP is becoming your ATP. Can you see it over here? Then that becomes empty. So all this circle will continue. As your proton gradient is maintained, the ATP synthesis will continue making the ATP continuously. Okay. So finally ATP is released. Clear? So with all these different small figures, I just want to convey the message is that, and please look it over here, the rotation also, gamma. I told you, right? It is rotating. So you can see the direction also over here. It is moving from one alpha beta to another to the another. So open, loose, tight, open, loose, tight. So just rotating and ATP is synthesized. Okay. Ranjan, is it clear to you?
Yes, ma'am.
Okay. So this is the same again, you know, just so that it becomes more clear. So another figure is there. Now, if the question comes, since it is 3 rotations are there, right? Always remember, now the question, what kind of question will come is, see, in a circle, you know that it has got 360 degree, right? Total 360 degree. Now, in this case, we have got the, how many are there? Okay, I just made it wrong, but otherwise alpha, beta, alpha, beta, alpha, beta we have, of which alpha, beta together, and this alpha, beta together. So it means that it has been divided into 3, right? Like this. So whenever this F1 particle of the ATP synthase is moving, it's rotating, how much degree, by how much degree it is rotating? It is the 360 by third. Okay, so here it is around 120 degree rotation. So what exactly is rotating? It is your gamma subunit. So clear? So whenever you have got the open means nothing is there. When ADP plus Pi binds, it becomes loose, and when it again rotates, it becomes your T. That is the tight where ATP is there. And then it loses its affinity, it leaves the F1 particle, and then another ADP comes. So loose, open, loose, tight, open, loose, tight. So all this rotation will carry on. Okay. So now is it clear to everyone the mechanism behind ATP synthesis? Is it clear?
Yes, ma'am.
Okay. Good. Okay. So this is another topic. So that is V class pumps. So till now we were talking about the, so we talked about the P type and now just about the F type, and then we have got the V type. Now why it is called P type? Remember, because it was possible for phosphate analog to inhibit the entire P class pumps, right? Now why it is called F? Any idea? Any idea?
If not an FN party.
That is because of the nomenclature. Then V, it is because first it was found in the vacuole. Okay. Now where do you see vacuole? You see in the plants. Animals also, but small vacuoles are there. Correct. So it was found on the vacuole membrane. So what do you call vacuole membrane? What is the membrane present on the vacuole?
Tonoplast.
Tonoplast. Very good. Okay. So it has got the similar structure and function of proton pumping, which is very much similar to F class pump. Okay. Now the thing is that in the case of F class pumps, it was down the concentration gradient. It means what? From high concentration to low concentration of H plus. Okay. Then it is called down the concentration gradient. Whereas in the case of V class pumps, it is against the protochemical gradient. Okay. So it means what? Whenever something is going against the concentration gradient, it is going to use ATP. Okay. So it has got the two structural domains. Okay. All these you have to remember. Okay. I'm so sorry, but bio means you have to remember. So once you understand, it is easy. Okay. That's why many pictures I'm putting, it is in your brain then. So once you remember the picture, you can remember everything. So it is again similar to F0 and F1. We have got V0 and V1 domains. So your V0 domains have got 5 subunits. What you have to remember is that V0 is responsible for the proton translocation. Okay. V1 domain is responsible for ATP hydrolysis. Just remember in the case of F class also, F0 was responsible for proton transformation, proton translocation, where F1 was responsible for ATP synthesis. Here it is hydrolysis because we are going against the proton electrochemical gradient. Now, where do you find these? You find on the membranes of the vacuole, on the lysosomes, endosomes, macrophages, osteoclasts, and renal intercalated cells. Okay. Now, can you tell me what exactly this pump will be doing if it is concentrating H plus? Concentrating H plus, what does it mean exactly the function of V class pump? What exactly it is doing? Any idea? I'll give you one hint. If lysosome is there and it is concentrating your H plus, what does it mean? What exactly it is doing?
pH it is maintaining.
Yes, it is actually decreasing the pH. Okay, so the pH is decreased. Now, in the case of lysosomes and endosomes, if nobody knows what is endosomes, it's the early stage of lysosomes. Okay, so what happens is like there are many proteins, like many enzymes are there which remain inactive at cellular pH. Okay, that is your around 7.2 to 7.4. So at that physiological pH, the enzymes are inactive. Now, when according to the requirement of the cell, if the proteases are needed to be activated, then what happens? The lysosomes will start decreasing the pH. Okay. Now, decreasing pH means becoming acidic, and these proteases gets active. Okay, and that's how many of your enzymes, like during our hunger, gets activated. So you feel hungry. I remember once I told you feeling hungry means that acidity burning sensation is there. It's all because of your V class pumps, because they're also called H plus ATPases. Okay, so they are the responsible pumps which are activating our enzymes in the stomach. What kind of enzymes we have in the stomach for digestion?
Ghrelin,
And
Pepsinogen.
leucine,
what Ranjan?
Pepsinogen. Pepsi.
Can you just write in the chat? Voice is not clear.
ghrelin and leptin, ma'am.
Okay, and what else? Those are for the fats. What about proteins? That is for the fat digestion. What about the protein digestion?
Pepsid.
Very good. It is the trypsin. So trypsin is present as trypsinogen. Okay. Then we have got chymotrypsin. Heard of it? So all these are present in the inactive form. So once the H plus ATPases decreases the pH of the stomach, these enzymes becomes from trypsinogen to trypsin, chymotrypsinogen to chymotrypsin. So they become active in order to carry out the protein degradation. Okay, good. So this is the structure. So if you see the structure of the V class pump, you see that it is very much similar to your F class pump, right? So this is similar to your F0, which is called, sorry, F1, which is called V1. And in the transmembrane region, we have got the VZ. So how many subunits are here? So around 5 domains are there, okay? And here 8 domains are there. Can you see structure over here? Again, alpha, beta, alpha, beta, alpha, beta is there, right? It is similar to the your F class pump. Now, only difference what you can see is that instead of H plus going in this direction, it is going in this direction. Can you see your H plus is there, it is going in this direction, making or decreasing the pH. Just remember this, man. You don't have to draw and learn the structures of these proteins. Nobody is going to ask. Okay. Just remember the functions and what all the important points are there. Now, so this is important to remember. So plasma membrane V80pases are involved in the process such as homeostasis and tumor metastasis. Now this is important because pH homeostasis, as already we discussed, okay, just to maintain. In the case of tumor metastasis, now why it is important? Because if in future you want to target any one kind of cancer, okay, so you need to know all the enzymes or the pumps which are responsible for helping your tumor to grow. Now in the case of tumor, what happens is you have got tumor cell is there. Now the tumor cell is highly functional. It means that if your normal cell is utilizing 2 glucose per second, for example, the tumor cell is utilizing 10 glucose per second. It means what? Maximum of your glucose taken by the body has been taken up by the tumor cells. Now more the glucose will be utilized, what will happen? All the metabolites will be produced and the H plus is also made. So more energy will be made. So everything is going for your tumor cell and then it starts metastasizing. Metastasizing means it starts getting spread. Okay, so it enters the blood system and then it moves to the different parts of the body, and that is the time when it becomes very difficult for doctor to, treat the patient. So we'll study this also in detail. Now it is also present in the acrosomal membrane of the sperm, which acidify the acrosome. So you know that sperm is there during the fertilization. At head region, the acrosome is there. So over there also these ATPases, V80pases are present, which activates the proteases required to pass through the plasma membrane of the egg, and hence the fertilization takes place. Okay. Now they're also present in the osteoclast plasma membrane, pump protons onto the bone surface, which is necessary for bone resorption. That is, the calcium, it will be resorbed. Then they are also present in the intercalary cells of the kidney, in the urine, allowing for bicarbonate reabsorption into the blood. So can you see, even though it's one kind of ATPase, but it is performing many roles. Now last, it is also present in the variety of biological process such as toxin delivery, viral entry. Now this is important because this is how viral make use of their ATPases. So they make they acidify the cell, host the cell, and then they enter inside the host cell and starts replicating. Okay, then targeting apoptosis, membrane targeting, sorry, apoptosis, regulation of cytoplasmic pH, proteolytic process. Just now we have discussed about the trypsinogen and chymotrypsin, acidification of intercellular systems. They all are the important roles of VATPases. So this is something, important slide which you need to remember. Okay. Okay. So this is what I was talking about in the case of the cancer cell. So you can see that over here. So when endosomes bud up from the plasma membrane, weak class bonds increase the acidity of the endosome. So what is happening actually? So let us say this is the example of your cancer cell we are talking. Okay. So what is happening? So the cancer cells is not following all your what do you say, all your cell cycle checkpoints, and has overgrown all this checkpoints of the cell cycle. Okay, it has become the tumor now. Now what is happening is that, can you see over here? One of the subunit has been expressed more as compared to the normal. Okay. Now this is nothing but a signal that your VATPase will be expressed more. So what is happening now? I have VATPases overexpress. Overexpression means from the normal high value of these proteins are expressed. Now you see that in the case of cancer cell, what they have done is they have increased rate of glycolysis is there. Okay, so I have got the H plus in high concentration, which is like acidifying your cellular pH, correct? Now this can lead to apoptosis, but this has been stopped or inhibited because of this H plus is actually taking up inside all the H plus inside. So lysosome is taking inside, and what it is doing, so inside this you had the inactive proteases, they got activated. These activated proteases now with the help of the exocytosis has been transferred outside. So outside, when the proteases are released, what they do, they start degrading the extracellular matrix or the other cellular tissues which are present over there. Okay, so is it clear? Should I repeat it again? Is it clear? So I'll say once again. Okay, so in the cancer cell, what happens is one of your subunit of your VATPase has been overexpressed. Okay, that is more than required. It has been overexpressed. So what is happening now? You can see that this is the A3 isoform. Now in the cancer cell, you have got the increased rate of glycolysis. That means increased rate of H plus will be there. Now if suppose the cytoplasm has got high H plus, it means the pH will decrease. It becomes acidic. The function of the the function of the cell will be lost. In order to avoid that, lysosome will take all the H plus inside. Okay. Now, increased H plus leads to apoptosis, but this has been ignored or this has been inhibited by these H plus. So all the H plus will be pushed outside or taken up by the lysosome. If the lysosome takes all the H plus, all the inactive proteases will become active. These active proteases are then, with the help of exocytosis, will be thrown out. Now these active proteases will finally start eating all your extracellular matrix or degrading the nearby cells. Okay, so this is how tumor cells make space for themselves and the nutrients available. So even if you don't feed the tumor cells with the nutrients, it will start killing the other cells, and the other cells, when they die, all the nutrients will be taken up by the cancer cell. Okay, is it clear?
Yes, ma'am.
Okay, interesting, right? I mean, while teaching itself, I don't feel sick now. Okay, so human diseases. This is one more because one of the question has come. So just remember in this. So what are the human diseases associated with the V class ATPases are osteoporosis. It is, sorry for the spelling. It is osteoporosis. Bone fractures due to brittle bones. So you get the bone fractures also, and that is the osteoporosis. Okay, bone fractures because it has become brittle. Since there is no calcium resorption, right? Remember we studied that it helps in the bone reabsorption. So osteoporosis happens if there is a mutation. Now, distal renal tubular acidosis. In this case, the pH of the urine goes below 5 and it becomes very difficult for the patient to go for urination. And the third one is X-linked myopathy with excessive autophagy. So in this case, the muscle cells are because the muscle tissues are becoming very weak and the person is not able to hold his or her own body. Okay, because the muscle cells are undergoing autophagy. That is, they are eating themselves only. So it becomes very weak and they cannot walk, move. So they use wheelchair and all. Okay. Yes. So I would like to stop over here. Thank you for listening.